which is indicative of group B strep, or negative is no color change. Okay, this is the second part to the Unit 2 lecture. In part 1, we talked about the different species of Streptococcus ACA and Enterococcus. This video is going to be more, we're going to talk about testing that is performed on strep species. So you'll see a difference from the testing that we talked about regarding staphs. You know, there's certain tests that are more appropriate for certain organisms, or if you suspect certain organisms, you're going to perform those tests over others to try to prove the presence of one organism. So we're going to talk about strep testing. We're going to talk about upper and lower respiratory tract cultures and pathogens that could be recovered from them. Then we're going to talk a little bit at the end about direct microscopic smears. So picking up at testing for strep species. Okay, the first, and we talked about these tests, or we mentioned them as we were going through the different strep species. So we're just going to go a little bit more in depth about the testing now, and some of these you will actually get to perform in class. So the first one is the optogen susceptibility test, also known as the P-disc. This is used for identification of Streptococcus pneumoniae. So if you suspect a certain isolate recovered from a culture is strep pneumo, you could perform a P-disc to confirm that. So the test is used to determine the effect of optogen, which is the reagent in that test is ethyl hydrocuprine hydrochloride. So that reagent, this is a disc test. It's one of those little tiny filter paper discs that are impregnated with the reagent. So the reagent in this one is ethyl hydrocuprine. So the effect of optogen on an organism. So optogen lyses pneumococci, so that's considered a positive result, but alpha strep are resistant, or that's a negative result. So what we're doing, and I'll go forward to the picture of the actual test results for just a minute. So we're using an inoculating loop to pick up a colony, so just one single colony from our original culture that we suspect of being strep pneumo, and then we're going to streak it onto our plate that we're going to perform our P-disc on. So just a very heavy streak back and forth, side to side, up and down, so that we get a very heavy inoculum of the suspected organism to the plate. We use forceps or tweezers to place that drug disc. So they come in like a little tube kind of. So you pick up one of those drug discs, place it in the center of that area that you inoculated. It has to incubate overnight. So about 18 to 24 hours of incubation. You come back and you're looking for that zone of inhibition or zone of no growth. And remember, I mentioned when we were talking about the organism, where some tests, any zone of no growth is considered a positive result. Some tests, you have to measure that zone. So the P-disc is one of those tests that you have to measure that zone. And we're looking for a zone that is 16 millimeters or larger. That's going to be our positive result. Again, every test that we perform in the lab has to have positive and negative controls, and those are, we set those tests up in addition to our patient samples just to ensure that our test is working properly. So our positive control organism is strep pneumoniae, our negative control is strep pyogenes. So that positive result would be that zone of no growth of 16 millimeters or greater. Negative would be no zone, or anything less than that 16 millimeters. Okay, the next test is the bacitracin susceptibility, also known as the A-disc. We use this one for the identification of group A strep. So used for the presumptive ID and differentiation of beta hemolytic group A strep, which would be susceptible versus other beta hemolytic strep species. We talked about this one in Unit 1 also, because you can use this test to distinguish between staph and micrococcus, but In this unit, we're talking about it in regards to identification of group A strep. So this one is performed, and I think we looked at this exact same picture previously. It is performed in the exact same way that that P disc is performed. So we choose a colony from the original patient culture, an organism that we find, or colony that we find suspicious that we think might be group A strep. And we're doing that very heavy streak on the plate, placing the A disc in the center of that heavy part of the streak. And what they've done on this plate is they've done the positive and negative control. So you would do your positive and negative controls in addition to, you would have another plate that had your patient specimen on it. And we're looking for, we're trying to prove the presence of strep pyogenes or group A strep. So that organism is our positive control. Our negative control is strep A galactiae or group B strep. This is one of those disc tests where any size zone of inhibition is considered a positive result and positive for the identification of group A strep. No zone of inhibition or the organism grows all the way up to the disc is considered a negative. So you can see right here around the A disc is that little thin zone of no growth. So all of this that looks kind of yellowish or lighter, and it's because it's beta hemolytic, it looks that way, but all of this is the organism. So it's growing in all of these spaces here, but not right up against the disc, versus group B strep, which is our negative control. Again, all of this is the organism that they've streaked, done that heavy inoculum onto the plate, but it has grown all the way up to the disc, which means it's resistant to bacitracin, whereas group A is sensitive. Okay, the next one is called the CAMP test. This is one that we use for identification of group B strep, and it's named after the people who developed the test. Use it to differentiate group B strep from other streptococcal species. So the principle is that group B strep produces what they refer to as a CAMP factor that enhances the lysis of sheep red cells by working synergistically with the beta lysine that's produced by Staph aureus. So synergistically means that they work together to produce this result. The positive control is group B strep or strep A galactiae, because that's the organism that we're trying to prove with this test. Negative control is strep pyogenes or group A strep. And we looked at this picture previously also. So you have to set it up with your Staph aureus control organism. You're doing a streak of that up and down the center of the plate. Your positive control is your group B strep. So you streak it right up to the edge of that staph without actually touching it. Do the same thing with your group A. Do the same thing with your patient, and then incubate, come back the next day. And what you're looking for is that arrowhead of hemolysis. So the organism is hemolyzed, the red blood cells in the media, but it's worked synergistically with that Staph aureus to produce this arrowhead of hemolysis, which group A strep will not produce. The bile-esculin slant auger, so this one we use for presumptive identification of Enterococcus and organisms in the group D or viridans group. So it will differentiate Enterococcus and group D strep from non-group D viridans. But remember, I don't know if you remember from the first lecture, I said if we're trying to prove the presence of Enterococcus or Enterococcus differentiated from group D, this one has to be performed in conjunction with the salt tolerance test, which we'll talk about it in just a second. So bile-esculin, it's a slant auger, so it's the same like media that's in the auger plate, like your blood auger plate, but it's poured into a tube on a slant. And what you have to do is you're going to pick up several colonies from your patient sample with your loop. You stab straight down into the auger, all the way to the bottom of the tube, and then as you're pulling the loop out of the auger, you kind of spread it across the surface of that slant. This one gets incubated overnight, and the next day you're looking for a positive result, which would be that the slant, the auger turns black. No change in the color of the auger is negative. So our control organisms, Enterococcus faecalis is our positive control. Negative would be E. coli or Strep pyogenes. Okay, and again, we're doing this in conjunction with the salt tolerance test. So this test is used to determine ability of an organism to grow in high concentrations of salt. So it can be used to differentiate Enterococcus from non-Enterococcus and to differentiate Enterococcus from group D. Enterococcus, the organism Enterococcus, can withstand a higher salt concentration than other gram-positive cocci, so it will grow in the presence of this increased salt. And if you remember, so that's our bile-esculin slant auger on the left with a negative result on the left, positive result on the right. This is our 6.5% sodium chloride or salt tolerance test, which is a liquid broth. We inoculate the broth, inoculate the liquid. It'll incubate overnight, the same amount of time that your bile-esculin does. And when you come back the next day, you're looking for cloudiness, which is an indication of growth, which means that the organism is able to grow in that increased salt content. So that is a positive result. The clear or no change in turbidity is negative. So again, positive control, Enterococcus faecalis. Negative control is Strep bovis. Positive is cloudy, negative is clear. So when we're using these tests together, again, we perform them together, we use them to differentiate Enterococcus from group B strep. Enterococcus is positive for both. If you have an organism that is only positive for the bile-esculin, but it is negative for the sodium chloride, that is a group D or non-Enterococcus strep. Okay, and the last test that we're going to mention that we would perform on strep species is the PYR. So we use this for presumptive identification of group A strep and or Enterococcus. Both organisms are positive for Enterococcus. So the principle, again, this is a little disc test. Impregnated discs serve as the substrate to produce alpha-naphthalamine, which is detected in the presence of DMCA by the production of a red color. So this is a disc test, but it's not one that we have to do the heavy inoculum and incubate overnight. This is one we smear a little bit of the organism on the disc, and we're looking for that color change after we add the reagent. So positive control could be either Enterococcus faecalis or Strep pyogenes, because both of those organisms will give a positive result. Negative control is group B strep, and again, your positive will result in a red or pink color change on the disc. Negative is no color change. So one thing that I will mention about this, because you've got two organisms that could be positive for this test, you have to be careful. Generally speaking, Enterococcus is not beta hemolytic. So if you're testing a beta hemolytic organism, you're probably suspecting group A strep. However, Enterococcus can rarely be beta hemolytic. So if you've got a suspicious beta hemolytic colony that you perform a PYR test on and it is positive, you then have to differentiate, is it group A or is it Enterococcus? If it's a non-hemolytic organism and it's PYR positive, you will know that it's Enterococcus because group A strep is always beta hemolytic. However, again, if the colony that you're testing is beta hemolytic, it's PYR positive, you have to have another means to differentiate whether it's Enterococcus or group A strep. But we just talked a few slides ago about the bacitracin disc test. You could perform that one to differentiate, or they also make like strep kit tests now that test for multiple strains of strep, so you could use one of those as well. One final test that I want to mention briefly, we talked about it as we were going through talking about the different species of strep, but there's not a slide for it here under the list of tests, but the hippurate hydrolysis test is used to differentiate group B strep from other beta hemolytic streps. It's a liquid broth in a tube similar to the 6.5% sodium chloride, where a positive result will result in a deep purple color, so that's indicative of group B strep. A negative reaction is no color change. Okay, the second portion of this Unit 2 lecture, we're going to talk about upper and lower respiratory tract cultures. So just a little bit of anatomy regarding the respiratory tract. So the respiratory tract is divided into upper and lower, with the upper respiratory tract consisting of all the structures above the larynx, so up in this direction, and the lower respiratory tract following the airflow below the larynx through the trachea to the bronchi and bronchioles, and then into the alveolar spaces. The respiratory tract and GI tracts are the two major connections between the interior of the body and the outside environment. So with, sorry, let me back up just a minute. So functions of the respiratory tract: performs respiration. So with that process, you've got exchange of oxygen and carbon dioxide. It delivers air from outside the body to the alveoli where that gas exchange occurs. And it also helps provide defense against respiratory tract infection or pathogens. So some host defenses. So the respiratory tract has many different types of defenses against infection that inhaled organisms have to be able to penetrate before they can cause disease. So some of those host defenses include nose hairs, which help filter air as it passes through the nasal passages; humidification, which causes particles to swell, so it makes it harder for them to enter the upper respiratory tract. Our normal flora helps prevent colonization of pathogenic organisms. Coughing and sneezing helps aid in expulsion of particulate matter. Mucus holds particles so that they can't pass further into the respiratory tract, and it also contains immunoglobulins, specifically IgA and other antimicrobial substances that fight off invaders. And then macrophages, which I previously mentioned, are the cells in our immune system that ingest bacteria if it's able to reach the alveoli. So evaluation of normal flora, host risk factors, and true pathogens. So there is a list in your book, a list of the bacteria normally present in the respiratory tract that have the ability to become pathogens. So those would be considered opportunistic pathogens because they're there, but they could pose a problem in immunocompromised patients. Reevaluation of pathogenic organisms. So also keep in mind that organisms that are considered respiratory pathogens must constantly be reevaluated as new relationships between bacteria and disease states are recognized. And then risk factors and true pathogens. So it's also imperative that you're able to distinguish between normal flora and pathogenic organisms by taking into consideration the quality of the specimen, certain risk factors associated with the patient and the organism recovered, because some are considered pathogenic regardless of the quantity. So Mycobacterium tuberculosis, which is the organism that causes tuberculosis, would be an example of one of those that no matter the age, the immunogenic quality of the patient, no matter those things, Mycobacterium tuberculosis is going to be considered a pathogen if it is recovered. So virulence factors that you have to take into consideration with pathogenic organisms. So these are things that enable microorganisms to proliferate and cause damage to the host. So adherence is one of those. So bacterial pathogens will attach themselves to host tissue. This is done by microbial surface molecules such as fimbriae, which we talked about in Unit 1. toxin elaboration. So certain organisms exacerbate toxins depending on the specific activity of the toxin and the specific cell within the body that it interacts with. Host evasion enables microorganisms to proliferate and cause damage to the host. One example of that would be chlamydia. It is an intracellular parasite, so it's taken up by the host's cells, but then the host cells protect the chlamydial cells from the host immune system. So they kind of have a way to hide out from the host immune system. Strep pneumoniae and some other organisms, they form capsules that prevent phagocytosis. So we've talked about that previously. Okay, some of the pathogens that are associated with upper respiratory tract. So this next section is going to be broken down into upper respiratory tract pathogens. So those are the organisms that can cause upper respiratory tract disease, and then the actual diseases that these organisms cause. So the pathogens, group A strep, we've talked about it. It's the most common upper respiratory tract pathogen. Haemophilus influenza type B. So this is bacterial. This is different from the influenza virus. Bordetella pertussis. This is the causative agent of whooping cough. Corynebacterium diphtheriae. This one's not as common in the United States and most of the developed world because of the diphtheria vaccination. Diphtheria was more common in children. The bacteria produces a toxin that causes what's referred to as a pseudomembrane that forms and can cause trouble with patients being able to breathe. The toxin can spread and be absorbed into the bloodstream and can cause organ damage. Strep pneumoniae, so we talked about this one as part of our normal flora, but in immunocompromised patients, it can lead to infections and diseases. Otitis media is one of those that it causes. And then there are many other viral causes of upper respiratory tract disease. So some of the diseases, pharyngitis, which is just sore throat. Group A strep is the most common cause. Sinusitis is an inflammatory reaction in the nose and sinuses. Viruses are more commonly the cause of sinusitis. Otitis media is an infection of the middle ear, most common in children. Strep pneumoniae and Haemophilus influenza are the most common causes. Epiglottitis that's the structure made of cartilage at the opening of the trachea that protects the airway from secretions and food when you swallow. So the condition can be rapidly progressive it's an infection of that structure and the adjacent tissue. And so that can result in obstruction of the airway. So this can be life-threatening, can be caused by the organisms Haemophilus influenza, group A strep, MRSA, and Streptococcus pneumoniae. Pertussis, otherwise known as whooping cough, it's highly contagious respiratory illness transmitted person to person by aerosolized droplets. It's caused by the organism Bordetella pertussis. The infection can last 6 to 8 weeks. There is a vaccine available, though we've seen more increases in cases the past several years due to folks not being vaccinated. And then laryngitis is just hoarseness or loss of voice, usually associated with the common cold or influenza. So then the lower respiratory tract pathogens and diseases. So some of the common pathogens, Staph aureus. So this is normal flora, though, so it would have to be present in significant quantities and with other clinical criteria. For instance, is the patient immunocompromised? Streptococcus pneumoniae causes pneumonia. Haemophilus influenza can cause pneumonia. Mycoplasma pneumoniae causes walking pneumonia. Mycobacterium tuberculosis is the causative agent of tuberculosis. Legionella pneumophilia. This one's, a lot of times it's difficult to diagnose because they don't gram stain well, they don't grow easily, but it causes Legionnaire's disease found in aquatic sources, often like air conditioning units a lot of times. And there was an outbreak of this one several years ago at the Mountain State Fair which took place during the summer months, and they had some of those like misting spray units that you could walk through, you know, hot day, and you're walking through this like mist of cool spray, like water spray. So it had become contaminated, and a lot of people got sick from that one. Bordetella pertussis and Bordetella parapertussis, Chlamydophila pneumoniae, and then again, a lot of viruses that are known to cause lower respiratory tract infections. So those were the organisms. Here are the diseases. Bronchitis is a common condition in about 10 to 25% of adults where there's excessive mucus production that can last for years. Cigarette smoking, infection, and inhalation of dust or fumes can be contributing factors to the chronic condition, while the acute is usually related to some type of infectious process. It's most commonly caused by viruses. Bronchiolitis is an inflammation of the smaller bronchiolar surfaces, an acute viral lower respiratory tract infection, usually occurring in children under the age of 2. RSV accounts for 40 to 80% of cases. The most common causes are due to viruses. Influenza. So again, this one is the influenza virus, not Haemophilus influenza, the bacterium. So influenza is caused by either influenza virus A or B. Other viral respiratory tract infections, the book goes into those a lot more, but we won't really get into them. Acute pneumonia, sometimes referred to as community-acquired or hospital-acquired pneumonia, is similar to bronchitis, except that there is lung tissue involvement. Most common cause is Streptococcus pneumoniae, and usually only when the strain is particularly virulent and the host defenses are compromised. Tuberculosis and other chronic pneumonias, so you'll see inflammation and accumulation of fluid in the lower respiratory tract involving the lungs' airways and those supporting structures. The major cause of illness and death, or it is a major cause of illness and death, most commonly caused by Mycobacterium tuberculosis, Staph aureus, and fungal elements. And then finally, empyema, which is a collection of fluid and pus in the pleural cavity, so in the area around the lungs. Often occurs in conjunction with pneumonia. The most common cause is Streptococcus pneumoniae, but it can also be caused by Staph aureus. Talking about some different types of upper and lower respiratory specimen sources. So those are broken into sterile and non-sterile specimen sources. So sterile, we have the BAL or bronchoalveolar lavage, where a bronchoscope is passed through the mouth or nose into the lungs. A large volume of saline is squirted into a small part of the lung and then collected for examination. You've got a lung biopsy where there's a piece of tissue taken for examination. Transtracheal aspirate, a small plastic catheter is inserted into the trachea through a needle that was previously inserted through the skin. This one is not a common procedure, but it is good for recovering anaerobes if anaerobes are suspected. It also reduces the likelihood of contamination by upper respiratory tract flora or diluted with additional fluids. And then a bronch wash or aspirate. This is collected by inserting a small amount of sterile saline into the bronchial tree, withdrawing the fluid. But the specimens could be contaminated with upper respiratory tract fluid. So then your non-sterile sources, sputum. So this is a specimen that comes from deep within the chest. So it can be expectorated, which means that it's coughed up by the patient, but it can be contaminated by saliva and the mouth on the way out, or it can be induced, which means that respiratory therapy department is going to help coax the specimen. A throat swab. So this is what would be collected if the doctor suspected group A strep. So just a sterile swab that is used to swab the back of the mouth. And then a nasopharyngeal swab. Those are the type that are used for flu and COVID testing, where they stick it in your nose. Okay, some special types of respiratory media that could be used. So most respiratory cultures would routinely only be cultured on your blood agar plate your McConkey or your EMB plate and your chocolate plate, and usually that's sufficient. But if there are other specific organisms that the doctor may suspect, specialty media might be necessary. So Leffler media is used for recovery of Corynebacterium species. So it's a selective medium containing serum. Lowenstein Jensen is used for mycobacteria. It's an egg-based agar. It's opaque. It's a solid medium. Bordet Genjo potato infusion agar for isolation of Bordetella. It's supplemented with glycerol, peptones, and horse or sheep's blood. And then BCYE, or buffered charcoal yeast extract. This one is used for isolation of Legionella. So some of these organisms that just might be a little more finicky and require some additional growth requirements might require some specific media. So quick review before we go into this last little section on direct exams. Which organism is the most common cause of upper respiratory tract disease? Group A, strep. The organism that causes walking pneumonia, Mycoplasma pneumoniae. What media do we use to recover Legionella? BCYE. Okay, last little section here is on direct microscopic smears or gram stains and then throat cultures. So direct exams, which can also be gram stains. So these are direct exams, come directly from the patient and go directly to the slide for observation. So what you're seeing on the microscopic slide might contain tissue, cells, bacteria. And again, just like the gram stain when we talked about it earlier, the purpose of this is to predict what should grow on the plates. It helps the direct treatment for the physician because we can get that direct smear or gram stain result out, 12 to 16 hours earlier than the actual culture preliminary results will come out. So if the doctor's seeing an indication from our gram stain result that says, that indicates a certain particular bacterium, then they can go ahead and start treatment. So correlation with growth. So you can kind of think of it as a means of QC, because if we don't grow out on the plates what we report from the direct exam or the gram stain, then something's wrong. We need to maybe investigate that. Okay, sputum, Bartlett's classification. So you use the gram stain to determine if the sputum sample is a true sputum that came from deep within that patient's chest, or if it's just spit. So it's done on low power. The criteria for the Bartlett's classification for sputum, and Bartlett's classification is for sputum samples only. You cannot use this criteria, this quality criteria, for any other type of sample other than a sputum. So it says that fewer than 10 squamous epithelial cells, greater than 25 white blood cells on low power field. So you have to have fewer than 10 epithelial cells, greater than 25 white blood cells per low power field. And then you would quantitate the bacteria on your oil immersion. So the squamous epithelials, the white blood cells, get quantitated or get reported from the 40x objective. Then you would go up to your oil objective to quantitate your bacteria. And just make sure white blood cells should stain pink. So if you're seeing lots of purple, this could mean that the sputum is either smeared on the slide too thick, and if that's the case, then it can't decolorize properly. Okay, for wounds, if you're doing a direct smear or a gram stain of a wound, you want to do report and quantitate bacteria and your white cells. And bacterial morphology, I spoke about this in Unit 1 a little bit, can indicate whether gram-positive organisms or staph are strep. So again, that could help the physician determine treatment. So if we report out gram-positive cocci in clusters, that's indicative of a staph infection. If we report out gram-positive cocci in chains, that's indicative of a strep infection. Those are going to be treated differently. And then lastly, and again, correlation with growth. So gram stains and your direct exams would correlate with what ends up growing on the plate. So you don't want to report out gram-positive cocci from the gram stain and then have gram-negative rods growing on the plates. If that happens, you would need to go back to that gram stain that was reported out the previous day and look at that again. Okay, your throat screen and throat culture. So your direct strep test, which is also known as a rapid A, this is the quick kit test. So these are the ones that, you think you're sick, your throat hurts, you think you might have strep throat, you go to the doctor or the urgent care. These are the little kit tests that they're going to perform. So the sample gets collected on a swab. You're testing for group A strep, which if you remember is never normal flora. And then this rapid test reflexes to a culture if that quick test is negative. So your kit test, which is usually done in 5 or 10 minutes, if that is negative, there is possibility for false negative results in those kit tests because it might be too early to detect, or we might not have gotten enough organism for that test. So a negative result from the kit test always reflexes to a culture. So that same swab, you've still got a throat swab, you're going to inoculate that to your blood agar plate, but then you have to, it has to incubate. So whereas you can report out those kit test results in 5 or 10 minutes, you would have to wait the following day to look at the growth on the blood agar plate. So collection, ideally, for group A screens, you want to collect 2 swabs, one for the plate, the other for the rapid test. And if that quick test is positive, then you're done. It doesn't have to reflex to the culture. It's only if it's negative that you're setting up that culture. For sputums and for bronch washes, the specimens will be delivered to the lab in a sterile cup, and the gram stain culture is set up from that specimen that's in the cup. Your media that this would be set up on for the throat culture where we're looking for group A strep, that only gets set up on the blood agar plate because the group A is the only organism that we're looking for. If it's a sputum culture or a bronch wash or something just beyond where we're looking only for group A strep, then that's where you add the chocolate plate and either the EMB or the McConkey. Modified Thayer-Martin is another plate we can add, but we would only do that if the physician is requesting that we're looking for gonorrhea in addition to any other respiratory pathogens. So when we evaluate hemolysis, again, when we talked about the streps, we said the hemolysis pattern that the organism displays can give us an indication of what organism we may be dealing with and kind of directs the testing that we would perform. So alpha hemolytic, you can see that with colonies of Strep pneumoniae. Beta hemolytic could be Staph aureus, could be group A or group B strep. Non-hemolytic could be Haemophilus influenza. So again, you're looking at that hemolysis pattern to help you determine what pathogens you could potentially be dealing with and what testing to perform. So some of those pathogens, again, group A strep, Haemophilus influenza, gonorrhea. Again, you would only be looking for gonorrhea when it's requested in cases of sexual abuse and other infections in adults resulting from oral sex. Staph aureus, yeast, and gram-negative rods. When they're predominant, gram-negative rods can be normal flora in the respiratory tract, so you would only work them up as a pathogen if it was the predominant organism growing. And then Streptococcus pneumoniae, again, that one can be normal flora, so it would have to be predominant or in an immunocompromised host. So just a couple review slides before we finish out this part 2 lecture. Purpose of the direct exam: helps direct treatment for the physician and correlates what should grow on the plates. What is Bartlett's classification? We want to see fewer than 10 epithelial cells and greater than 25 white cells. And again, this is only for sputum samples, and that the epithelials and the white cells get quantitated on 40x objective. Everything else, or the bacteria, gets quantitated on oil. Okay, that's the end of the lecture 2 for unit 2. In the next video, we'll get into Neisseria and Moraxella.